Welcome to the Watson Lectures at Caltech. Yeah, come on. I hope everyone here had some time to spend outside today. Now, not just because I heard that the activities were pretty fun, but because this is Earth Week. As a community, we're celebrating the birthday of the modern environmentalist movement. And I think it bears repeating that the first Earth Day in 1970 grew out of bipartisan government concern over the impact that humans were having on the environment and that environmental pollution was having on human health. They specifically chose the day, April 22nd, because they wanted to maximize student involvement in these discussions. And so they picked a day that was after spring break and before finals. Like, they knew they still had a captive audience, you know? And since then, millions of students have participated in Earth Day activities. And with that, I want to acknowledge all of the students in this audience, especially those from Blair High School who were this month's Junior Watsons. And they had a chance to tour the Karthikeyan lab and talk to researchers. Yeah, amazing. And, you know, learn about what it's like to do some scientific and engineering research at Caltech. And I hope they also told you that Caltech faculty have been researching the Earth's climate and environment and ecosystems since long before Earth Day made it cool. Tonight's speaker, Dr. Smruthi Karthikeyan, continues that grand tradition of ecosystem research. And in her work, she looks to harness the power of microbes to improve the health of both humans and the environment. She's going to spill the tea on the secret lives of superbugs, but before she does that, you're going to learn a little bit more about her as a person. You'll hear an introduction from the division chair for Geological and Planetary Sciences, and you'll watch a short video that will tell you a little bit more about her. After her talk is when you get a chance to ask your questions. So we'll have a little Q&A. What will happen is a QR code will appear on the screen. Take a picture. It'll take you to our online platform where you can enter in the questions that you might have for your speaker. If you don't feel comfortable doing that and you prefer old school, then just flag down one of the ushers. They'll have a card. You can write your question, and then they'll make sure it gets uploaded to the platform so that I can ask your question. We'll answer as many as we can. And then afterwards, you're invited to join us for tea and coffee and hopefully a little bit more conversation. So I will ask you to look at your phones now, silence your phones, your watches, any other technology that might become disruptive. And of course, note your nearest exit. And help me welcome to the stage the leadership chair for the Division of Geological and Planetary Sciences, Dr. John Eiler.
Thanks so much, and welcome all of you to yet another wonderful Watson Lecture night. It's really delightful for me to be able to be here to introduce Smruthi, who has only been my colleague for about 2 years, but is already a beloved part of the campus and both the engineering division and geological and planetary science division. You might wonder, what am I doing up here introducing a proper engineer? That's the Caltech way. We are all each other's colleagues. We are all each other's friends. Once you're inside the walls, it's all the same. So it's a great pleasure to be able to introduce her. So Smruthi is a fascinating scientist in a whole bunch of ways. For starters, she is just an incredible presence, very energetic. You're going to learn that yourself in just a moment. Knows virtually everything about virtually every branch of science you might want to bring to bear on interesting problems. And it couldn't come on a better day. So what better day than Earth Day to have a person who is studying the environment in every way you might imagine. The environment of the ocean, the environment of the soils, the environment of you and your interior. So a perfect match of the person to the moment. We're going to learn a bunch of things about the ways in which Smruthi has been exploring I think of it as like a vision after talking to her of the way in which the microbial world interacts with, transforms all of the environments around it, including ourselves. We are an environment for the microbial world. And this brings her into many different problems, impacts society in many different ways. I think it's this concept that scientists are guided by some kind of distinctive, unique vision is an important idea. It tells you something about scientists, that they are not just executing a plan that somebody else wrote. They have their own ideas. What's something that's unusual about Smruthi's case, and there's really no way to dance around this, is her inspirations, after talking to her for a bit about this, all seem to involve poopy water. So don't laugh, because you are part of the poopy water problem. So from the beaches near her childhood home, which were unsafe to swim in, the Gulf of Mexico following the Deepwater Horizon oil spill, the hunt for COVID infections in sewage in the UC San Diego dorms. I guess she was going after COVID and, I don't know, leftovers of Doritos or something from a dorm sewer. The teeming microbial ecosystem in our bodies that is the cause of a lot of our health and sickness. All of these are one thing to her, and she takes subjects that are to us impolite dinner conversation and turns them into scientific gold. She does this by merging biology, chemistry, physics, and really explores the way that these three different, seemingly different branches of science interact with each other to create an environment. And she approaches it as a genuine polymath with tools of DNA and other omics, environmental chemistry, field work, going out in the ocean, and on and on and on. So I think you're really going to enjoy all that she has to show us tonight. And now let me turn your attention to a video by the Watson Lecture team, which will introduce you to Smruthi in her own words. So thank you, and I hope you enjoy the evening as much as I know I will.
I grew up in the south of India in a place called Madras or Chennai. It is a coastal city. I grew up a mile from the beach, but the water quality was really bad. It was basically unusable. My mom was a teacher, and my dad's a CPA. They couldn't help me with my schoolwork or anything, but they definitely encouraged me to pursue whichever STEM field I wanted and explore other options too. Every year I used to pick up a different interest. So I wanted to be an architect once, I wanted to be a designer once, I wanted to be a person who just does calligraphy, because why not? And then I was gifted this book, The Magic School Bus. And it was basically just science with like a cool lizard. That's kind of where it started. I'm Smruthi Karthikeyan. I'm the Gordon and Carol Trevique Assistant Professor and the William H. Earth Scholar at Caltech in the Engineering and Applied Science Division. My lab works on developing computational and experimental techniques to study how microbes interact with each other in their natural environment. There was not enough infrastructure for the environment where I was growing up, and I felt like that was one area that really needed to be strengthened. I found chemical engineering to be one of the closer fits because I could transition to environmental engineering. I started at Columbia, and then I did my grad school at Georgia Tech and did my postdoc at UC San Diego Medical School. The professor I worked with at Georgia Tech, he was one of those few professors who just used to tinker around the lab just because he wanted to do science. And I told him what I wanted to do, and he personally trained me in the wet lab. So my idea was to look at how these microbes in offshore oil spills can actually be leveraged as a predictive modeling tool to say how long an ecosystem will take to recover and how we can harness this to invigorate the natural or indigenous microbial community to clean these up. A lot of the techniques I developed for looking at microbes in the environment are very different from how typically clinical work is done. So at that point, I switched to the medical school because I thought that a lot of these unconventional methods could be harnessed to look at microbes in our own body because the host is an environment in itself. We had this idea where can we go a step forward and basically do clinical disease epidemiology based on the genomic signatures we got from wastewater, saying did a person in one building have a party and then give it to everyone else, or did they all just travel and each of them had a different source of the virus? So we were able to go to that level of resolution from a single environmental sample, which then became the mainstay for San Diego County and how they approached infectious disease epidemiology. So the most exciting thing about Caltech when I started was the amazing intellectual diversity in terms of research. Caltech actively fosters collaboration. You have people at Caltech who are senior faculty who are basically the pioneers of their field, and it is so easy to reach out to them, work with them, and collaborate with them, which I don't really see in any other setting. My students span pretty much all divisions, and they talk to a person in a completely different lab in a completely different field. They'll brainstorm these ideas, and the next day I'm talking to their PIs, and then we're writing up an actual proposal to test these ideas out. I already work with people in biology, biological engineering, chemistry, chemical engineering, geology, and planetary sciences. When you're young, you think of all these scientific questions in your head, and you have these wild hypotheses. And it's very exciting that Caltech actively encourages to test out a lot of these ideas. And I felt like a lot of good science comes from having these interactions. My episode of the Magic School Bus would be probably called Fantastic Microbes and Where to Find Them, and it would feature Miss Frizzle and her trusty lizard hopping from one environment to the other and looking at how microbes there evolved and live, or how they've made their living. Microbial species outnumber the stars in our galaxy, but we don't really think about it at all. But I think we can basically look towards the microbes to answer a lot of our questions.
Hi everyone, thank you for being here. I'm really excited to be here. So I hope to take you on a fascinating journey of microbes all the way from our sewers to the human gut. So let's dive in. Microbes, the fascinating world of microbes. I call them the tiny titans of Earth. So they were here for most of Earth's history, for about 90% of Earth's history. We were there for a tiny, tiny, tiny fraction of it. They made the Earth, which was sterile, more habitable. They created oxygen, which is what we need now. And as you can see, we are multicellular organisms. We started somewhere over here. Mammals and humans. So a long way. So the fascinating thing about microbes is that they have somehow found a way to live in any habitat imaginable. So let's dive into some of them. So here you can see microbes in Antarctic sea ice, which you would think is not a very hospitable environment for microbes. Here you can see an oil droplet, and you can see microbes surrounding the oil droplet and breaking them down. So this is a microbe that we isolated from the Deepwater Horizon oil spill, which breaks down crude oil. And here you can see like a bunch of microbes, which are called fettuccine rocks, because you can see how they're sort of twisting around. So these are found in hot springs. So all of these you traditionally think are not very hospitable environments, but they've clearly found a way to survive in all of these environments. And not only that, they found a way to thrive too, not just live. And this is why it is very important for us to look at the incredible microbial diversity on Earth. Mainly because looking at how these microbes evolved in such extreme environments give us a clue as to how we can search for life on other planets, because a lot of these extreme environments are analogs to Mars and other planets. They are the main reason for sustainability on the planet. We use their gene products for biotechnology. Also, a lot of infectious disease epidemiology is done through microbes. So when we look at the microbial diversity, it is vast. They are one of the most diverse creatures on Earth. And let's take this giant ship, for example, and this tiny ship. What we currently know in traditional microbiology by culturing or growing our microbes and studying them is basically this tiny boat here. But the actual majority of our microbes on the planet are so vast, and we have to look for other techniques to be able to study them. So we use this term called microbes in the wild, mainly because microbes are social creatures. They don't live by themselves. They do need other microbes to sustain them, either because they can mooch off resources from them or just because they like being as a community. So you can just see here, this is the microbial community in a lake in Michigan. Sewage sludge, which is really gnarly, so you can see how diverse they are. And this is just your human tongue. So this is actually our very own turtle pond at Caltech. So you can see the incredible diversity of microbes in just the turtle pond here. And all of these have light-reflecting pigments. So when excited with UV light, you can see the different colors. And same turtle pond, you can actually see them in motion. And you have a bunch of different microbes, so you can see how they're closely spaced together. So now when we talk about microbes, usually when you think of a microbe, you always think of it in a, say, clinical or hospital setting, like E. coli, where you try to grow them in culture and then you try to analyze their traits or characteristics. But unfortunately, that only represents 5% of the actual microbial diversity on Earth. A majority of the microbial communities in our natural environment cannot be cultured, mainly because they need other resources that help them grow. So that's why we call them the uncultivated majority. I also like to use a tongue-in-cheek reference, calling it the microbial dark matter. It's not exactly how you would think of it in physics. What we call dark matter is what we can't see. If we can't culture them, we think they don't exist. So there's a lot about the microbial world that we don't know. So through the talk, I'll go over how we can look at the dark side of the microbes. Okay, so when you think of the tree of life, it's evolved a lot from when Darwin first proposed it to what we think of the tree of life now. So this was around the tree of life in about 1969, which was a big deal back then. So when you look to construct the tree of life, you basically grow these microbes and you look at them and see what their color is, what their shape is, and if they're more similar, you put them together on a tree, much like a family tree, and you assume they're more closely related. So that is how this tree was built. So you can see bacteria here, which is like a very tiny portion. So all of this changed with the advent of computational biology or nucleic acid sequencing. What that shows us is, instead of having to wait for these to grow, we can bypass any of this, and we can use sequencing or computational biology to actually reconstruct the tree of life without ever having to grow a single bacteria in culture. So this was started when Carl Rose, he actually pioneered the idea of looking at nucleic acids and using nucleic acids as a proxy to construct family trees for microbes. So what he did was he discovered, by just looking at the nucleic acid signature, there was another form of life, which was called archaea, which predates higher organisms, which is us. And this was because these could never be grown in culture, and they were basically found by sequencing. And initially, they were called Martian-like bugs, mainly because they only grew in really extreme environments, and they were actually found in Yellowstone National Park, which is not exactly a very habitable environment. So now that we know how the old tree of life looks, this is currently a later version of the tree of life. This is still outdated because it's 2016. So everything you see here with a red dot means they were generated computationally. They were not grown in culture. So you can see a majority, we've basically missed this entire section of bacterial phyla because we've always depended on growing or culturing microbes to look at them. But with the explosion of computational biology, we've basically expanded the entire tree of life. So every red dot you see shows that these were all novel or uncultured bacteria. And it's fascinating because a lot of these are very ancient. For instance, archaea evolved before eukaryotes. It's just that we didn't know that they evolved before because we didn't have a methodology to actually find that out. So how do we look at reconstructing genomes? So think of it as a jigsaw puzzle. So genomes are made of strands of DNA. And let's say you take a million-piece jigsaw puzzle, and then you just shatter it into millions of pieces. So there are two ways. if you have a picture of what the jigsaw puzzle is supposed to look like, it makes it easier for you to put it back together. So think of this as your genome, which is just shattered, or your jigsaw puzzle. In this case, let's say this is your jigsaw puzzle picture. So we know this is the sentence. So all of your genome pieces are like this. So you assemble them. So you know how your sentence is. So you take all of these pieces, and you basically align them to your reference. So this is called genome assembly. So this is how you assemble a genome when you know what you're looking for. However, what if you don't know what you're looking for because you have, say, 1 million different species in a gram of sand or a gram of soil? How do you do your assembly then? So let's say you found an article. You printed a whole bunch of it, but you did not read it. You just decided you're going to print a bunch of it. Since you didn't read it, you don't know what it said. But then you have a cat which just likes blowing things up, and it blew the entire stack of your articles. So now you have a bunch of pieces. So you didn't read it, so you don't know what it said, right? So now comes your assembly. So you don't have a reference. It's like assembling a jigsaw puzzle with no picture. So how do you do this? So you have all of these pieces. So what you try to do is you're trying to see if you can find overlaps. Because you have multiple different pieces, you try to find overlaps, and you try to assemble them. And I'll give you an example. So it's called genome assembly. So this is a very mathematically complex problem, but I've simplified it here. So let's say each sentence is one read of your genome. So usually, you're not going to have 6 reads. You're going to have 6 million, 60 million, 6 billion reads. So now you have this sentence that really doesn't make any sense because they're just strands. So now you have to find a way to put this together so it actually makes sense. But you don't know what this sentence said to begin with. So what do? So we do something called a graph theory algorithm. So there's a lot of math in this, but I've tried to make it very simple. So what you do is you're trying to see if there are any overlaps. So here you can see these two are the same, if they have the same color. So what this does is it tries to rearrange your reads, each of this is a read, as a node, and an overlap as an edge. To help you picture this, think of it as a subway station. So you have your nodes, which are basically your stations in a subway, and then you have your edges, which are basically your tracks. So the more sequences you have, the more complicated your graph becomes, right? So we take all of this, so now we organize it to make more sense. So now you know, see, these two match, these match, these match. So using these, you try to trace them back, and you try to put together the jigsaw puzzle. Again, this is only 6 reads. You will have millions and billions when you're dealing with genome sequences. And then once you have them, you can rearrange them and make them a single sentence. So this is how genome assembly works when you don't know what you're looking for, or you have a cat that just blows a bunch of stuff up. This is how you try to put them back together. So what is the importance of all this? So let's say I have an environmental sample, right? Which is billions or millions at least, or hundreds of thousands of microbes. The same thing I told you about sentences happens in a genome. So you have multiple copies of a genome, and then you shatter it. It is called a process called shotgun sequencing. It is called shotgun sequencing because it is done much like the random firings of a shotgun. So you shatter it into 1 million or 1 billion pieces, and then you find overlaps, and you assemble them to give you a larger segment of DNA. And we do this a lot for environmental samples. So let's say you take a gram of sand, or you take anything. You take a bunch of microbiome from your cat, or your dog, or the turtle pond water. So the diversity is incredible. So if you take a gram of sand, you're going to have millions of different bacteria. This is just a small representation. And then you're then going to break them up, like I told you. You shatter them into millions of pieces, which are DNA. And then, like I told you, you assemble them. So you find overlapping segments, and then you put them together to make sense of which microbe is there. So once we do that, you can construct whole genomes of all the microbes. So like putting together a jigsaw puzzle, let's say you had 100 different microbes, You're basically developing algorithms that'll put 100 different genomes without ever having to grow them in culture, but we're doing them computationally. So let's say these are 3 different microbes. I'm just showing you a small subset. Your diversity is much more. Let's say each of this represents different genes in a genome of a microbe. You can take them together and construct family trees by looking at how similar your genome is, so how similar the alphabets making them up are, and put them together and create family trees. But we can do a lot more than just that. So let's take one of these microbes, for example. Just by looking at their genomes, we can tell what environment he lives in: water, sand, soil, oil, whether he likes oxygen or not, what kind of energy or food he likes. So these are just basically genes. So we develop algorithms that will go in and look for information. It makes sense of all the 4 alphabets that make up a genome. And then, this is cool. So we want to take this a step further and see, can we go and look at evolution in bacteria by looking at their genomes? So for instance, can we say which bacteria he exchanged genes with? Who does he like associating himself with? Does he like associating himself with any other bacteria? What viruses he was infected by? And what antibiotics he's resistant to? So these are some of the questions we're exploring in the lab, and we combine experiments and computational algorithms to see how microbial communities become resistant to antibiotics over time, especially in areas like wastewater as well as the human gut. So I want to say this is a double-edged sword. With a lot of data comes a lot of noise. So you would have heard a lot about the advances in the field of microbiome. So a lot of microbiome fields will tell you, they'll look at your poop sample and tell you what diseases you have, what diseases you can get, or what diseases you can solve. However, a lot of this is done with extensive amount of computational data. And what they do is, for instance, let's say they take a cohort of a person who has type 1 diabetes. Another cohort who does not have type 1 diabetes. And they look at the microbiome by sequencing your poop sample. And then let's say they find microbe X in a person with diabetes. And a person without diabetes does not have microbe X. So they come to the conclusion that microbe X causes diabetes. Again, there's a lot of signal or noise, so I would offer a cautionary tale. Just because you see a microbe and a person has a disease X does not mean they're correlated. However, unfortunately, when you rely only on your computational algorithms to predict your fate of a microbiome, this is what happens. And as you can see, there were studies that said there's bubonic plague in the subway. The New York subway is nasty, but it's not plague level nasty yet. So that's why we really need to come up with a better way of verifying what our computer algorithm predicts, especially when you're dealing with such big data. Okay, so this is what our lab does. So you would have probably met some of my students here who make up my lab and do all of the cool research, and also made all of this, most of the slides you see today. So what we do is we try to see how microbes interact amongst themselves and their environment, and how we can harness those for both human health and environmental health. So we develop both computational and experimental methods to visualize microbial interactions in complex environments like wastewater, soil, or the human gut. Okay, so I mentioned antibiotic resistance before. So this is a really big public health threat at the moment, and it's only getting worse. So by 2050, the deaths from antibiotic resistance will outnumber the deaths from cancer. So it is a very dire situation, and this is mainly because bacteria have evolved over time to basically resist any of the new antibiotics in the pipeline. Also, we haven't made a new antibiotic in forever, so we're still counting on the ones from decades ago. And with antibiotics, as you can see here, by 2050, you can see 70% would be the mortality for people 70 and older due to antibiotic resistance. So when we traditionally think about antibiotics, we think about hospital-acquired infections or you fall sick, they give you antibiotics, you take a lot of it, and then you become resistant. So when we always think about antibiotic resistance, that's what we think about. Oh, we take a whole bunch of antibiotics, or we misuse antibiotics. However, it's a little more nuanced than that, because antibiotic resistance are not just in hospitals. They move through a lot of microbial ecosystems, right from the human gut to wastewater to natural environments which have had no prior human influence at all. So this is how, let's see how an antibiotic resistance works, or how antibiotics work. So let's say this is your bacterial cell. Antibiotics are of different classes. They're of different classes on how they act. For instance, let's take penicillin. So you have your bacterial cell wall, and then they're made of peptidoglycan. So basically, your penicillins will attack that. So once your cell membrane, it basically keeps all the cell stuff inside. And once you lose the integrity of the cell membrane, it basically disrupts your cell functioning. So that is how some of them work. And some of them attack the ribosomes. The ribosomes are cellular machinery that makes proteins. So it stops protein synthesis. It can also affect your DNA or RNA synthesis steps. So this is how most of your antibiotics in different classes work. And the problem is, as you use more and more antibiotics, they've somehow evolved to resist them. Oh. So like I mentioned before, antibiotics are not just in hospitals. They've been in environments where there's no human influence. In fact, all of the antibiotics currently in use were from soil bacteria. They were produced as secondary metabolites. So those microbes used them as defense mechanisms from other predators. But that is pretty much where all our antimicrobial products came from. The latest antibiotic, which was discovered recently, came from a sandy soil in North Carolina, from uncultivated bacteria. So that's how uncultivated bacteria are an untapped source of natural products. And this just came a couple of weeks back. So another new antibiotic that kills drug-resistant bacteria was actually discovered in a lab technician's garden from his soil. So this is now the new pipeline for antibiotic discovery, looking back at soil or the environment. So I know some of you are already familiar with some of these microbes because you would have taken a personality quiz that matched you up with one of these pathogens. So these are called escape pathogens. They're called escape because of all the names, and also it's a very tongue-in-cheek reference because they are resistant to antibiotics. And these are flagged by the CDC and WHO as the really nasty ones in hospitals, which they find really difficult to treat because over time they've developed resistance to most antibiotics. So you can see a bunch of them, like Klebsiella causes pneumonia, but also is there in some gnarly urinary tract infections that just won't go. And E. coli is, everyone knows about E. coli. There are a lot of pathogenic E. coli, but there are also a lot of E. coli that are basically harmless. So recently, they found escape pathogens at the ISS. ISS is the International Space Station. So you can see this is the ISS area, and all of these colors you see are how much bacteria they found in the International Space Station. So they're pretty much there everywhere. And when we think of antimicrobial resistance, like I mentioned, there are a bunch of different hotspots. You have soil, because soil, you have a lot of animal influence, you have a lot of antibiotics there too. And then you have the human gut, which has its own selection mechanisms. And then you have the most nasty of all, the wastewater system. The wastewater system aggregates human fecal matter, it has veterinary waste, it also has agricultural runoff, all of which have pesticides, herbicides, antibiotics, and anything you can name. And also indigenous microbial community that just live there. So to give you a little bit of an idea of how wastewater is very important in infectious disease epidemiology, I'll take you a little bit to where I was a couple of years back. So I graduated my PhD during the pandemic and moved there during the pandemic. So this really giant toilet is a metaphor of what happened to my life back then. And then I moved to San Diego County. So at that point, San Diego County had a lot of infections, and they were not keeping up with their tests. So a lot of people were looking towards wastewater. Though SARS-CoV-2 is a respiratory pathogen, you still shed it in your gut. So these blue dots show the viral load we saw in San Diego County's wastewater, and the red shows the clinically reported cases of SARS-CoV-2 in San Diego County. So you can see a really good correlation with wastewater giving you a leading indicator. So up to 10 days before wastewater can predict how your caseload is going to go in the county. So if you're not familiar with San Diego County, it's a large county. So what you see here in orange or yellow are the sewer lines that connect San Diego County. They have 3 wastewater treatment plants. This Point Loma is the main one, which is a snapshot of 2.2 million people. All of their waste join in this one wastewater treatment plant, which is what the data you see is here. So at the same time, we were looking at UCSD. When we were at UCSD did not close their campus. We had 25,000 people living on campus, and they wanted a way to use wastewater not just in a large scale, but can we use it at a single dorm building to bypass clinical testing and still keep campus open. So that's when we came up with the method to take the SARS-CoV-2 RNA virus from wastewater, separate it from all the junk, and see if we can say how much COVID load is there in wastewater. So again, you see San Diego is a very large campus. So it has about 360 buildings. So we put about 130 of these robots. These robots sample wastewater from a manhole, which drains from each dorm building in UC San Diego. UC San Diego is a very large campus. So every orange box you see there represents a UC San Diego building, residences as well as non-residence or research buildings. And if you see a yellow dot, it means there was an automatic wastewater sampler in that building. So every day we used to process these samples, and if you see a red light up, it'll automatically ping your cell phone saying that that building had a positive in wastewater. If you see a blue, it means that there was no positive in that building in wastewater. You can click on each of it and see how long a positive stayed in that building. So in order to see if this actually worked, when we first started the program, every single person on UCSD campus was required to test via clinical swabs every week, just to make sure that the wastewater captures every case that clinical captures. So we found that not only wastewater captured every single case that the clinical swab captured at a fraction of the cost, but it picked up 85% of the cases early, mainly because a lot of people are young and they never had any symptoms, so they didn't think they had to test. So you can see a really good correlation between the wastewater positive samples and the clinical samples. So at that point, we just swapped the candy in the vending machines to nasal swab tests, so students would just swab. And if you test positive, you go to isolation, everyone else goes back and lives their life as usual, so none of the buildings were shut down. So when you think all these I showed you were just looking for presence-absence, it's called qPCR, which is the same nasal swab test, which basically tells you if you have COVID or not. You can't really say what variant you have, or do you have a new variant? Do you have an old variant? Or, is out of 1 million people, what if one person has a new variant? Can you still pick it up in wastewater? So at that point, almost all wastewater studies were just presence-absence, or how much of the viral levels were. And when you swab your nose, you only have one variant. When you have wastewater, you can have any number of the thousands of different variants at any different proportion. If there are 10 people with Delta, 1 with Omicron, and maybe 2 with some exotic variant, can we pick that up from one sample? So to do that, ignore all of the math. All this is showing is it's a computational algorithm that picks up from wastewater all the different variants and at different proportions. We called it Freya. So we took this, and then to test if it does a good job, we combined all the human nasal swab samples where we know which variant was already there in different proportions, and we asked our algorithm to predict what mixes were there. It did a really good job, and then we thought, okay, let's try it on real samples. So we compared 31,149 nasal swabs from San Diego County with 1,200 wastewater sequences, and our algorithm found alpha and delta in San Diego County 11 to 14 days before the first reported case in the county with just a fraction of the samples. Not only that, the first alpha report in San Diego was the first one reported in the US and was picked up by our wastewater sample 11 days before the first clinical presentation. And we found the very same sample then in the clinical testing a bunch of weeks later. Same thing in UCSD campus as well. It could predict the peaks much before it happened. What you see on the top are wastewater samples. The bottom are clinical or nasal swab genome samples. Not only that, it could pick up variants never seen in any clinical area. So it picked up a bunch of variants which were never seen in any of the clinical samples. And it predicted mu, which was a new variant, even before the WHO or CDC classified it as a variant of interest, which really helps us pick apart these different variants and where there are blind spots in clinical testing. And same thing with Omicron. Before the very first Omicron in the US was detected, we picked it up in wastewater at a very small abundance a good 11 days before. Not only that, it could also say which was the next emerging variant. For instance, when Omicron came, we had this BA.1. But then our algorithm said that the next variant would be BA.1 with this new mutation, which was then reclassified by CDC as BA.1.1, and soon enough it overtook. And not just that, it could go, our algorithm in wastewater could predict not just a variant, but subvariants too. So you could go into such incredible detail. And you could also see which variants persist. Do some persist longer than others? And all of this was done with a wastewater sample. And currently, pretty much all the academic labs, the California Department of Public Health, CDC, FDA use these as their mainstay in all of their genomic surveillance programs in their national and international programs now. And we did the same thing on UCSD campus. So the closer they are, we know if there's an outbreak. So if you see a lot of clusters together, it means that a person threw a party in their dorm and then infected everyone else. If you see them far apart, it means that though they were in the same dorm, they picked up COVID from very many different sources, and they did not spread it to each other. Not only that, if a person in a dorm went and infected someone in dorm from dorm X to dorm Y, and then they moved to a different dorm, the wastewater could trace those signatures. So it could easily pick up, like, people here infected a person in this building, and they moved to this building, and it would have an exact clinical swab match. All the data are de-identified, so we can't identify the person from the wastewater sample, but we can tell how the spread occurred. And now we made the tool open source and public, so even a person with no computational biology background will be able to use the source. And this is currently done in San Diego County. So it now can predict which would be the next variant, how fast it's growing. So this is still a little old, but it can track data in real time. All of this is from wastewater. It precedes clinical, and you can see this particular variant has a higher growth advantage. This one is probably coming down. So I hope I've convinced you how incredibly powerful wastewater genomic epidemiology is. So all of this was during COVID, and we wanted to, currently in our lab, we want to see if we can push this further and study how bacteria evolve. Can we use wastewater as a place to see bacterial evolution in real time? And that's where we kind of segue from a real pandemic to a more silent pandemic, the antimicrobial resistance pandemic. Like I had mentioned before, for antimicrobial resistance, something called close contact is very important. So when you think of antimicrobial resistance, again, you think, oh, I had a whole bunch of antibiotics for an infection, and now I'm resistant. That's how you usually think. Or you think of it in a clinical sense, where a person gets sick, and then they get resistance. But there's a lot more. I hope I can convince you that antimicrobial resistance is a lot more than just that. So in wastewater, you can see you have a lot of close contact of microbial communities, which is very similar in the gut microbiome too. So the closer the bacteria are, there's a higher chance that they can exchange genes, which is what I'll be talking about. So usually in clinical settings or when you take antibiotics, how it happens is resistance happens when your prolonged exposure to antibiotic. And then all of these cells, some of them get mutations over time because they're seeing antibiotics. And then as they propagate, they then spread the infection to the other cells. And over time, they propagate, and then they get antibiotic resistant. And this is just one set of bacteria. But what if there are a lot more bacteria in an area? How does this happen? So one of the main ways antimicrobial resistance spreads is through something called horizontal gene transfer. So when you think of gene transfer, you think of passing genetic material from your parent to your offspring. In humans, that's how it always works. You pass your genes to your offspring. And that is how the information flows. Bacteria, they kind of seemingly violate this. They can share their genes with their peers. So if they sit around with their peers long enough, they can start exchanging their genes. So that is why it is called horizontal gene transfer. It is done through something called mobile genetic elements, MGEs. They're exactly what they sound like. They're mobile elements, which are genetic in nature. And we call them the agents of open source evolution. So they're basically chunks of DNA or RNA. They can move within the bacterial cell, or they can move between different bacterial cells. And this is one of the major ways antibiotic resistant evolution occurs. So this allows antimicrobial resistance genes to jump from one species to a completely different species, which was never a pathogen. And when we look at escape pathogens, we're missing this whole gamut of microbial communities who can still acquire these, though they're not quote unquote pathogenic. And the way they're done through agents called bacteriophages. Bacteriophages are viruses that attack only bacteria, unlike SARS-CoV-2, which attacks humans or eukaryotes. These only attack bacteria. Plasmids, they're kind of circular DNA, which can replicate independently of the host chromosome, and they can move around. Sometimes they're also called selfish genetic elements because they can mooch off the host. And sometimes they also provide an advantage for the host. And then transposons, or transposable elements, are called jumping genes. So these are very important, and Barbara McClintock actually won the Nobel Prize for discovering jumping genes. However, she discovered this for corn or maize, where she found that genes could jump within their genome and then provide different properties or appearances because they're moving from one area of the genome to another. A very simple analogy is you can think of your DNA. There's two mechanisms. You can cut and paste or copy and paste. So if you copy paste, it basically copies your transposon from one area of your genome to another. So now you have two copies at different locations. Or you can cut, and then your gene, your piece of DNA can move from one area to another. So now this confers a very different property to your genome. Same thing for antimicrobial resistance. It can basically move from one, and then it can be mobilized, and then it can go from one cell to another. So that's how they can jump across species. And they're done through three main ways. One is through phage. Your phage basically is your virus that sits on bacteria, and then it injects its DNA into the bacteria. And another way is called conjugation, which means it has something like a pilus, which is just fibers, where it forms a bridge between two bacterial cells, and it can transfer your genetic material. The other way is just you have DNA that's just floating about in the environment, and another bacteria just picks up this DNA and incorporates it into its own genome. So this is how they spread. So this is an example of a plasmid, which is a segment of DNA which can replicate on its own. So this is the pilus, where it forms a bridge. And then this is an example of a bacterial phage, which is injecting its genetic material into an E. coli cell. And these things happen pretty fast. So if you see here in this E. coli cell, you can see how it forms these bridges. And they form them pretty fast too. And so even if they're pretty far apart, they can reach out and then transfer their genes. So this is how antimicrobial resistance spreads in one of these areas. So when you look at biofilms where they're more smushed together, this makes it even more of a hotspot for genetic exchange. So you can see all of these come together in this sort of opening a Pandora's box. So all of these exist in our human gut as well as wastewater, which is why antimicrobial resistance spreads much faster in these environments. And this is very, very important in the environment because they can alter how your genome behaves. And if you see here, these were all the viruses we know before sequencing. And now you can see just by computational biology how much more we know about the viral diversity. It's massive. And this is why it's very important for us to have a very holistic view of antimicrobial resistance. And to identify these, it's very difficult computationally for you to identify who's a host, who's a mobile genetic element, where do they move from? Which bacteria do they move from to which bacteria? So to do that, we need a computational and experimental framework. And I'm not going to go into too much details. There are a couple of different experimental ways we do this, where we try to link the physically close cells together. So if you're a mobile genetic element, we try to see if they're close together, we will try to put them together or put their DNA together. And then, so here you can see this is, let's say, a plasmid which has an antimicrobial resistance. This is your E. coli. We try to put these two together to say which, who gave the gene to whom, or how did they transfer. And ignore all of this. All of this is just an early computation because imagine the complexity. We have 3.6 billion reads we get from a wastewater sample, and we have to tease apart these interactions. So ignore all of the computational. This is just the back end showing how much work goes in to get this to work. And then, again, it fits a model, which just says how well your computational algorithm is doing. The more straight the line, it means the model is doing better or doing really well. So we take that. So this is an example of a wastewater sample. So you can see the resolution of bacteria. So each one, you can see the proportion of bacteria. Even at 1% abundance, we can find them. And there are millions. This is from a single wastewater sample. So you can go, so you find some of your MRSA, and you find some of the other escape pathogens here in our wastewater, and you can find the abundance too. So anywhere from 0.1%, you can see the incredible diversity in wastewater. And using that, we construct a family tree. This is very important because you can see if a gene jumped from here to here, or here to here, or it jumped from this end to this end. So it gives you an incredible understanding of how far these genes can jump and why we're missing this incredible diversity when we only look for the same 4 or 5 pathogens. And then this is just a snapshot of an example. So I've taken 1, 2, 3, 4, a bunch of these from here, just a handful, and we found this one beta-lactamases. So these are resistant to penicillin, cephalosporins, and cefotaxime. So we found that about 6 of these were shared, or they moved between 6 different microbes, and they were not even closely related. So they jumped across different species, and they've shared their genes, and that's how they got antimicrobial resistance, because these are not bacteria you would usually think of as antimicrobial resistant. And same thing here. So if you see the escape pathogens, just from our wastewater sample, just from one sample, we can gauge which all antibiotics each of these bacteria are resistant to. So we don't need to grow them, wait for them to culture. This will give us an entire list of what bacteria which antibiotics they're resistant to. We can also look at their genes and say what is their mechanism of action. So when we get therapeutics, it's much easier to say, looking at this bacteria, we can say, oh, this has found a way to counteract this antimicrobial resistance in a certain way. So this is most of the escape pathogens, but you can see how many drugs they're resistant to. So wherever they're joining, it means they're resistant to that many drugs. And finally, we can also look at phage. So phage is just a virus that infects a bacteria. So you can see, I've just shown you one phage, which is basically has a host range that can infect a whole bunch of bacteria. So it means that it can transfer its genetic elements across a bunch of different bacteria, which is another way antimicrobial resistance spreads. And now with our methods, you can actually find these out from a single sample. Same thing, this is just how a phage genome looks when you assemble them. So you can look at each of these genes and say how they're propagating. So this is for tail protein. You can see how they can attack a bacteria by just looking. This is how a phage genome looks. All of these are genes, and each of them have a function which we can infer computationally. So one of the ways that we used them was we work with clinicians at UC San Diego School of Medicine who do phage therapy for cases where they have extensive antimicrobial resistance. So there was one case where they had an outbreak of XDR, means extensive drug-resistant Pseudomonas aeruginosa. So that Pseudomonas was resistant to all of these drugs they tested, and this was an eye drop-associated infection. They tried everything, including the last resort antibiotic drugs like colistin. Nothing worked. And then they were looking for phages. We found a couple of phages in wastewater, which our computational algorithm predicted that could work against these XDR Pseudomonas aeruginosa. So you can see the phages from wastewater here. And indeed, those were the only ones that actually worked in killing the Pseudomonas aeruginosa and was used in experimental phage therapy. So you can see the Pseudomonas, which is an escape pathogen. And you can see the blue, which is basically a phage infecting that bacteria. And now they're using it for cats. So if you don't like humans, I mean, they work on cats too. So this cat, this was recent, was saved by personalized phage therapy from a very bad antibiotic infection. So with that, I'm coming to a little bit of the conclusion. So you can see we're trying to visualize. Now our goal is to look at what our algorithm predicts and see if you can visualize. So here you can see E. coli cells. And you can visualize a phage infecting the E. coli cells. And you can see about 20, 30 minutes, they can take over the community really fast. And our hope is to actually use a lot of these algorithms we use for wastewater to look for alternative therapeutics for phage therapy. So you can look at very unconventional sources for clinical problems too. And same thing, I have a student who's now doing using this pipeline and adapting it for sepsis, because usually in a hospital when you have sepsis, this is very life-threatening and time matters. Usually they try to put a whole bunch of antibiotics and see which sticks, or they have to culture, wait for the culture to come back, and then see which they're resistant to. So we're trying to see if we can reduce the time taken from sample to analysis in clinical settings too, to see which antibiotics would work faster for someone who has sepsis. And the last part is looking at the same thing in your gut microbiome. So gut microbiome, much like wastewater, has a lot of close interactions. And we think that that is another area where antimicrobial resistance would spread. And we're trying to see how antibiotics work in the gut too, and at what stage we can see these gut bacteria acquire antibiotic resistance horizontally from a different pathogen. And that's currently one of their methods we're developing in the lab. So we're trying to add antibiotic to heavy water, which will separate. So heavy water is just an isotope of water, and your microbe, which is active in the presence of antibiotic, will take that label. Then we can separate that label, and only the ones that can grow or are resistant to antibiotics and are not sensitive to antibiotics will grow despite antibiotics. So we're hoping we can separate them and then find out which of the gut microbial communities will then acquire antibiotic resistance. And we work on this with a collaborator at Caltech, and you can see this is the instrument that will help us separate the antimicrobial resistant cells from the non-resistant cells. And we're just starting with this, and you can already see how it's, if it's CD, it means it's swapping its hydrogen bond with heavy water, which is deuterium. So if it's growing in the presence of antibiotics, if you see cells, it means that it is becoming resistant to antibiotics. And you can see this happen in about 24 hours. And hopefully, with that, I've convinced you that there's more to antibiotic resistance than just clinical settings. And with that, I want to conclude my talk, before which I want to thank all my students who have made this incredible work happen. I've only been here for about 2 years, so we're a fairly new lab, along with the private foundations and the federal agencies that support our work, along with Caltech and the Caltech Center for Environmental Microbial Interactions. Since it's Earth Day, I have a bunch of pictures of the students on the field, because we also do a lot of environmental microbiology work. Yeah, with that, I'm happy to take any questions.
Smruthi, your work is so complicated. My mind just absolutely boggles at the size of the data sets that you're working with. Putting the parts of your talk together, is it possible to take the predictive strategies that you were using for the SARS-CoV-2 and then look at how this antibiotic resistance is sort of the genes that are being transferred and how it's evolving in the bacterial populations you're studying, and make predictions so that we can get ahead of some of those evolving superbugs?
That's a great question, yes. So that would depend on the mechanisms. Like I mentioned, there are a couple of different mechanisms on how antimicrobial resistance spreads, especially in a complex environment like wastewater, where you have a lot of biofilms, and then you also have cells that are just floating about. So there are different factors. So sometimes if you have more biofilms or close contact, it means that there's more chance for your genes to spread. So if you have not as much close contact, but you have a lot more phages, then your mechanism is different. So currently we're trying to see if we can actually quantify each of these mechanisms and at what time scales they take place. So once we validate that, then it would be easier for us to build a more robust pipeline that can predict these, much like COVID.
I see. I feel like I have to get the obvious question out of the way immediately. So how is AI impacting the work that you're doing?
That is a great question. So I am not an AI researcher, so take whatever I say with a grain of salt, because that's outside of our area. So all of our computational algorithms are very mathematically based, not machine learning or AI based. So with life sciences, it's a little more complicated because, like I mentioned, with microbiome science, there's a lot of noise, and then we don't know what the signal is. So when you take AI or some other predictive machine learning tools, they can maybe help us with developing certain hypotheses or helping us filter data or make our life easier. But every hypothesis which is predicted has to be tested experimentally or validated, because some of the papers we saw which predicted, say, cancer microbiome, which all relied exclusively on machine learning tools, which would pick up, basically what we found was it was able to predict with about a 99% accuracy in a person looking at their microbiome, saying which type of cancer they had. But in reality, it was because the machine learning model was attaching itself to contaminants or background noise coming from the human or host cell, and it wasn't actually from the microbial signal. So which is why we really need to validate. If a model is too good to be true, sometimes it's because it is. We really need to verify it. So I'm not saying there's a lot of potential, but at least in life sciences, it really needs to have this feedback where, okay, you have a hypothesis, let's see if it actually holds up in the lab. And that's why we try to hopefully work, to sort of merge these two fields where it's a more feedback loop.
You mentioned in your talk that antimicrobial resistance might overtake, or is predicted to overtake cancer as a concern for human mortality. How can that be? Why are we moving so fast? How do we stop it?
That's a good question. So again, the burden is very different in different countries. So even let's take wastewater. So a lot of places don't have proper sewer infrastructure. So they're not treated, and they just go into your water bodies. So we noticed, even after wastewater gets treated and you do have effluent, we can still see some of these persist. So imagine if you don't have any sort of treatment, or you don't have a good enough treatment, and you just let it into a water body. It just has a lot of time to fester, and then it has input from agriculture or veterinary sources. So it has more time to also evolve. For instance, if you take Tijuana River in San Diego, at this point, it's basically mostly sewage, because when we were looking, we were testing the sample, it gave us the same levels of sewage as actual sewage. So that's not good.
As a UCSD alum myself, I find this highly concerning.
That's just because it's not treated, and it gets worse when you have sometimes with your rainfall where it brings in more into the ocean.
We're talking about how the evolution of this antimicrobial resistance is, damaging for human health potentially, but is there any way in which these, sort of evolving resistances could be beneficial?
So not entirely, if they're beneficial to the microbes, not us. But again, horizontal gene transfer is not always a bad thing, because we associated with antimicrobial resistance. So we saw horizontal gene transfer in action when we were looking at oil spills. So when we were looking at the Deepwater Horizon oil spill, you had a ton of crude oil that washed up onshore in the Gulf of Mexico. So when we were looking at the microbial community evolution, so microbes can degrade oil. So they can break down crude oil, which is carbon and hydrogen, into different components and make it more innocuous. So we found there are certain genes we can find in these microbes that can degrade oil. So they're called alkane monooxygenases. They're called so because they can break down alkanes, which are a component of crude oil. We found that when we started, only a couple of bacteria had this gene. But over time, because of crude oil exposure, they then started exchanging genes. So by the time we finished our experiment and when the oil was completely cleaned out, a lot of bacteria horizontally acquired these genes. So you get some specialty genes like these, which can be beneficial. So a lot of times when you have groundwater contamination or other sources of contamination, if they release, say, a new, like if there's an EPA Superfund site and they release some new chemical or a pollutant, initially it may be toxic, but over time we saw microbes evolve ways to actually break them down. And then it starts with maybe one microbe in a community, but over time, because they're exposed to it so much, they start sharing these genes, which actually help break down pollutants too. So they're not always bad. In antimicrobial resistance, it's bad. But also, you see antimicrobial resistance genes in like pristine environments, but they're used by cells as defense mechanisms or to combat stress in their own environments, or also metal resistance. So if you have a lot of arsenic, they have metal resistance, arsenic resistance genes, arsenate resistant genes too, as a plasmid, which they can then transfer to other microbes, and over time they've somehow found a way to evolve.
So you've mentioned disasters like Superfund sites and oil spills. Would the locations of the recent Los Angeles fires potentially be an area where you might find something new and exciting?
That is a great question. Yes, we some of the field sites that we actively work on also had fire burns. So it's one of our active areas of research where there are burn sites where we see how the microbial community is shifted, and if they've shifted, how are they more resilient or not? So those are, and we're also looking at how they can adapt to drought, especially in California's climate volatility.
Yes. We're always talking about drought in California. So what are some of the big questions that are still to be answered about the gut microbiome and its effect on our overall health and longevity?
That's a great question. So I know there's a lot of, I know gut microbiome has been related to every single disease. So I would take that with a grain of salt because we're still working on some. However, there are some areas of research that are FDA approved, like for treating C. diff infections. Fecal microbiota transplant is FDA approved, meaning they take the poop of a healthy person, and then it's taken by a person who has a C. diff infection because their microbiome cannot overcome this infection. So they hope a healthy person's poop will then reinvigorate their microbiome to basically overcome this infection. So that is one of the ones that's FDA approved. So with antibiotic resistance, there's a lot we still don't know, because when we think of gut microbiome, we only look at poop, but A lot changes by the time it goes through your intestines before it comes through poop. Especially with antimicrobial resistance, we want to see over what time scales do you get antimicrobial resistance, over what time scales, or what are the metabolisms that make some bacteria more susceptible and some not. So we want to see which of the microbes' metabolism is shut down by antibiotics, which of them can tolerate them, which of them are sensitive, and which of them are resistant. If they're resistant, it means they can actively grow. If they're sensitive, they can't, and they have some repressed metabolism. So these are some active areas of research we're hoping to find, so that'll help us better develop therapeutics. So for any successful therapeutics or application or translational development, it really helps to understand the fundamentals or the mechanisms of how it happens in the first place, so that'll help us better inform therapeutics.
Yeah, some of that work on poop transplants and the microbiome are so fascinating. A lab mate of mine, who I'm going to throw under the bus because I don't think she's here, Tegan Wall, once told me that her entire life would be different if she could just get the poop transplant from a skinny, happy person.
Okay, so there are some side effects because there were some unintended side effects for some people. So who took the FMT gained a lot of weight. That's not something they saw. For some people,
Teagues.
yeah. So it did. And also, there was a study where they took 2 mice, one lean, one obese, put them in the same cage, and then they saw over time, are they both going to become the same? They did, because when you put 2 mice in a cage, they'll eat each other's poop. And they do, so they ended up becoming.
I mean, this is really giving, this is extreme dieting, but I'm not, it's maybe a little too extreme. Well, thank you. Next month's Watson lecture will be the last lecture of the season. Can you believe it? We've made it all the way to the end. It will be a chief engineer from the Europa Clipper mission that Caltech JPL is doing. And so I feel like we have to end with a question about astrobiology and bacteria. You know, are we going to see like different types of evolution of, you know, this antibiotic resistance if we look on other planets instead of just the ISS?
Ooh, interesting. Well, the ISS is mostly because of humans living there, and their microbiome from the skin is basically just
imprinted. As usual, we are the problem.
Right, and it's also because they're there for a really long time. It's more important because you can't just escape and leave. So now what they're doing is they're planning to take these portable sequencers aboard their space missions and the spacecraft missions to see if they can sequence their surrounding environment really quickly and get their data really fast instead of having to collect all these samples, wait, bring them back to Earth, and then assess them.
All right. Well, thank you so much. Please, another round of applause.